Hi, everyone. It's great to be here. Thank you for coming. I hope you had a nice little break just now. So like Mike said, I use machine learning in my research. I'm going to be talking today about what we're going to call traditional machine learning. And so, Mike just kind of drew this contrast, and maybe you've even seen a figure like this earlier on today. But we use sometimes these terms, artificial intelligence, machine learning, deep learning, sometimes in the media almost interchangeably. But they do have more specific meanings and are kind of nested within one another. So artificial intelligence we think of as we are teaching a machine to mimic human behavior. My apologies for the video. Machine learning, more specifically, usually means some sort of statistical methodology that then enables the computer to learn through experience. And that would be also in contrast to deep learning, which has made huge waves in the media recently. So I will not be talking about deep learning. I'll just be talking about more traditional machine learning methods that you may even apply now and not realize that it's machine learning. So what is machine learning? What can we use it for? I have a toddler at home, so I kind of really like this comic. There's maybe 3 really broad types of machine learning. And first, I can sit with my toddler and I can show him a cube. And I can say, this is a cube, it has 6 sides. This is what it looks like, these flat faces. And he can sit there and say, yes, okay, cube. I'll remember that. I've now learned it. That's pretty ideal, but you get the picture. I can also just sort of leave him unsupervised to run around and do his thing and learn by experience. And so he might learn to classify things into categories like this is good to eat, this is not good to eat. And there might be some mistakes along the way. But hopefully nothing that lands you in the emergency room. And the third type of machine learning, and that might be more along the lines of the deep learning methodologies, is trying to synergize and make sense out of lots of different things and come up with new ideas. So these 2 first methods, this supervised and unsupervised learning, this is what I'm going to be talking about today. So supervised, unsupervised, I've given a little bit of a kind of intuitive overview. What does that look like in practice? So I'm going to use, as an example, 2 methods that we use in my lab maybe every day. And you might use as well, at least this first one. Who here has ever used principal component analysis? Anyone? Okay. So in principal component analysis, which you might have seen in papers, this is usually a great way to find structure in your data. I often use this as a kind of a first step when I get a complex data set. For example, in my laboratory, part of what we do is we study the secretion of immune cues and downstream immune signaling. And so I might use multiplexing assays to measure the levels of those immune signaling proteins in a sample, whether that is plasma from a patient, brain tissue from a mouse, anything like this. And at the end, I get maybe 50 of these molecular species, and for every single sample that I've looked at. And I said 50, but let's pretend it's 3, because I've got 3 dimensions in which to kind of give a better, more intuitive example. So I might have those 3 species. You can see this up here. Let's call them, proteins 1, 2, and 3. But it's not just molecules. You could literally anything. Hair color, eye color, height. Anything that you can put a number to. You could use this particular type of examining your data. And for each one of my samples, I would say, okay, in these 3 proteins I measured in this sample, x coordinate 1, y coordinate 1, z coordinate 1, and I put my dot. Each one of these dots might be a sample for me, or for whatever data type you have, you would similarly have your dot. And at the end, when I get through all of my samples, I might have a cloud that looks like this. And that cloud usually isn't completely random all over the space. Instead, it might look like, let's call this a really strange looking football. So I've got this kind of football type shape that is situated in these axes. And what a unsupervised linear machine learning method like principal component analysis might do is look for the directions of greatest variation in my data. Because this will tell me something. So for example, if I had these samples that were from mice, maybe some of those mice have a disease and some don't. Some are male, some are female. Some are young, some are old. And I might expect, depending on what those proteins are, that there would be, variation that might correspond with some of those variables. So I'm going to look for the structure. And what principal component analysis will then do is it will say, well, what is the axis, the linear axis, if I could draw a line through the direction of greatest variation in this weird-looking football, what would it be? And you know what an American football looks like. It has this kind of egg shape. It would be that long axis, and so it would draw a line. And then the next line it would draw would be the next greatest, largest axis of this football-like shape, and so on. And depending on how many things you've measured, here we've measured 3, you get 3 lines. Something like this. I hand drew this, so forgive me. These 3 axes are now your new axes. And you just kind of rotate it. And if you can remember back to high school math classes, what you might remember is that when you have a line that's a diagonal on 2 axes, you can describe it as a combination of those 2 axes. Y equals mx plus b. And so what we would have here is 3 axes, and so we would say, okay, this x prime 1, that greatest direction of variation, that is a combination that we can calculate of our original 3 proteins, and that would be a type of signature. And we might want to go back and look at what these samples are and try to explain to us what these or try to explain to ourselves what these new axes represent. And this would show us some of the structure in our data. Now, what if our objective is different? What if we were trying to specifically draw one of these axes down a line, for example, that would separate 2 groups in our data? These mice have a disease, these mice don't. What would we do? This is the idea behind supervised methods. So I can present the algorithm with my data set and say, here are these proteins I measured. And here are their identities, the identities of that sample. Here were the measurements, this one had a disease. Here were the measurements, this one didn't. Just like when I show my toddler the cube and I tell him it's a cube. So I tell them the answer, and then the machine will say, okay, this is great. I can calculate then where those directions should be. So my data might look something like this. And do I have a mouse? Yes. So these green dots here, maybe those are the mice with a disease, and the yellow ones are healthy. The colors don't look great up there, my apologies. These darker ones versus these lighter ones. Now, instead of, this is the same data, by the way, I just colored it. Instead of finding, like in the principal component analysis example, the greatest direction of variation in your data, it now knows that you're looking for something in particular. And it will take that into account. It will compromise between those two objectives. Find the greatest variation, separate these two groups. Tell me the difference between them. And so your axes might look a little bit different. And again, my apologies, I hand drew this. It's 3 dimensions and a 2-dimensional projection, but you get the idea. They're slightly different, and depending on your objective, may be better. I can tell you briefly why I use, for example, a supervised method often in my research. This exact one, actually, partially squares. And it's because often the difference that we're trying to discern between our two groups might not be the biggest variation among your subjects. This is especially true if you do human subjects research. In some of my previous research where I was trying to understand cell signaling in type 2 diabetes, the human population is very variable. And it turned out that the difference that I found, I would never have picked up with an unsupervised method because it represented about 8% of the variation in my data. But it was incredibly accurate at predicting these two things. And what are those others? It's just noise, for example. Things that you can't control, especially in human subjects. So supervised methods can be highly useful in noisy data sets, and when you're looking for something very specific, and when you're not sure how big that difference is going to be. By the way, stop me at any point and ask me questions and tell me I didn't explain something clearly, Shivani. Never a dumb question.
But if supervised data method is more specific and more selective, then why do we even use the unsupervised?
Good question. So the unsupervised methods are, we could also say they're unbiased. And they will show you the underlying structure of your data. For example, if you have a large data set and you're not sure, and you know there are a bunch of differences between maybe your subjects or whatever your N is, you might want to understand how those differences sort of fall out in space without telling the algorithm to look for one or the other. Sometimes there are so many of these differences that if I told it to look for one specific one, I'm ignoring the others. So for example, if I use the supervised learning method, and there are ways to get around what I'm about to say, but and I said, I want you to tell me the difference between disease and not disease. Everything else, it would try to project that onto that one axis, and all those other differences that might be interesting, male, female, age, etc., that might get fractionated among lots of other axes. So there's a lot of reasons that are specific to your particular question and research direction why you might use unsupervised. I like to, probably every data set that comes across my desk, I do a PCA first. Because I'm curious, unbiased, what does it look like? And then I might throw it up kind of like this and color it differently for all the variables that I think are there. And see how they fall out and see what made the biggest difference, especially if I'm doing something that nobody else has done before. And so I have no idea, actually, what's going to matter and what isn't. This might be especially relevant in cases where you're not sure if there's going to be a sex difference, for example. I know for a lot of us, the NIH now requires that we do everything, we address sex as a biological variable. And it's very important because it was ignored for years. But sometimes there isn't a difference and sometimes there is. And so what I always like to see first thing is like, is there sexual dimorphism in this data set? And if there is, that's super interesting and I can follow it up with a study that powers for that. If there isn't, then I can move ahead with kind of like just the cohort size that I have. So great question. Yes?
In the supervised methods, do we have only one variable that we compare, or can we have multiple variables that we can compare in supervised?
Fantastic question. Yes. So we can absolutely, I know that you guys had kind of a linear algebra primer earlier in the day. So these are matrix-based, they're linear algebra-based methods. And so in principal component analysis, you have one input to the algorithm, and that is a two-dimensional matrix that is basically, I measured these 50 things in these 50 samples or whatever. In, for example, partially squares, I have two inputs, and it's that first one, but then it's also in these 50 samples, this is their identity. The math does not care if that matrix is a 50 rows and one column, or 50 rows and 50 columns. Now, there are a lot of caveats to it that mean that people don't do this. I actually did this in my postdoc a lot, and it can be very tricky. And so you just have to make sure you really understand what you're doing and you really perturb the system. But absolutely, you can look at multiple outcomes. You can also technically, in that input matrix, instead of it being 2 dimensions, it can be 3. And yeah, I'd be happy to discuss that with you later. It's, again, trickier. But the math doesn't care how many dimensions it is. But you might when you're picking the number of latent variables and et cetera. But yeah. Any other questions? All right. Like I said, stop me at any time. Like, raise your hand, shout it out. Yes?
So this xy, x1, x2, x3, this may be just a concentration of certain proteins, right?
Yeah.
In a certain experiment. And basically, the green dots and yellow dots may be the cases where there is a disease and there is no disease.
Mm-hmm.
Right? And your goal is by looking at the data point on this cloud to say whether it has a certain disease by analyzing the concentration of these proteins, right?
That would be a goal, an example goal, yeah.
Basically to drive some kind of plane, which split these two sets in the best way.
Yes, exactly. Yes, and this can be for classification or it can be for regression. And I think I'll talk about that in a couple slides. Yeah. All right. So I'm gonna tell you about a few different types of methods. I'm not gonna go into a lot of depth. I just wanted to give a brief idea of some of the most commonly used unsupervised and supervised methods. And if you're really interested in one of these methods, I'm absolutely happy to talk with you afterward. And or I'm at the end of this, I have like all of my contact info. But I couldn't go by without telling you about some methods, but I do want to get to some of the other. So happy to answer general questions, but specific questions about specific methods, I'll talk to you afterward. So unsupervised learning, there's maybe two sort of general types. I could subcategorize this further, but we have clustering methods. So you have some data, and again, you're trying to understand what goes together. Are there, are there different groups? Are there sort of, or is it all just like continuous, flat plane? So many of you might have seen something like this, hierarchical clustering. You'll see it a lot in gene expression studies, for example. I believe this particular example is from knockdowns of tyrosine kinase and seeing what that does to the proteome. So this is a proteomics where each row would be a particular tyrosine kinase knockout, and each column would be a different type of protein that's expressed. And so what hierarchical clustering does is it tells you It puts these in groups, and you can see that this is almost like a little tree, dendrogram here, that will put these together by their similarity, branching and branching and branching until you can see maybe clusters that would make sense. Now, obviously, I'm not showing you actual, I mean, this is actual data from an actual paper, but I'm not going to discuss it. But you can see that in this paper, they found that certain ones of these knockouts tended to be more similar. And they happened to have grouped these in a way that they thought, oh, this makes sense. We now know that these groups of tyrosine kinases, when you knock them down, seem to do this type of thing. And you could go back and look at what these columns, what the proteins in these columns are, and be able to get some biological insight. This is just a different representation with not looking at the individual data, but just what's similar. So you may have seen these types of figures in papers. Another very common clustering method, this is probably more intuitive, k-means clustering. When you have data with a lot of parameters, you just see what group's closest. And the way this works is it sort of starts and it randomly, it asks you, how many clusters do you want? This person said 3. Knowing that there are 3 clusters here, they said, please show me where are the boundaries in these clusters, where are my groups? And so it found a point and it measured all the distances from the points and found the centroid and decided that here are their clusters. So k-means clustering is really great when you have a good idea how many groups of things there might be. And if you're not, there are ways to get around that, but this is a good place to start. So the second type would be dimensionality reduction. And this is where you have a data set with, again, a lot of different parameters that have been measured, and it's too many. So for example, if you do RNA sequencing, transcriptomics, you'll get 20,000 or so protein encoding genes. That's a lot. It's a lot. It's too much for you to look at all of them and try to decide. So what dimensionality reduction will do is that it sort of will choose which dimensions draw the biggest differences. And so I can give you, I'm not going to go through all of these, but probably the most familiar might be, whether you know it or not, is TISNE and UMAP. These are nonlinear methods, obviously. If you've ever seen a map like this is from single cell sequencing, or if you do flow cytometry, you use TISNE a lot to get the map and understand where the cell type populations are based on the different markers that have been used. As you can see, so this paper is from single cell sequencing of brain tissue. And one thing that these types of methods is really good for is, again, it looks like clustering because it kind of is. Dimensionality reduction often ends up being clustering. And here they've separated out all of these different cell types by their gene transcripts that they've expressed. Supervised methods. So again, they fall into kind of 2 categories. Classification is the first. This is nominally what I sort of showed you with the example with PCA and PLS, which is you have classes, groups, and you're trying to separate them. And you're trying to figure out what is the difference between these 2 groups, these 3 groups, these 6 groups, however many groups you have. So this is a method called decision trees. This is actually from one of my own papers, and it's a very simple decision tree. You've probably used a decision tree in your everyday life, especially if you have any clinical duties. If this, go here. Now make your next decision. Yes, no, okay? You've probably even seen these flow diagrams as memes or like whatever they are. In this particular case, we were trying to understand the difference between samples from those with HIV and those without, and based on their cytokine profiles from mucosa. And so this is based on just, let's see, we didn't actually even need that many cytokines to make these decisions. It's 1, 2, 3, 4, until we separated out all of our samples. Regression. So regression is honestly, algorithmically very similar to classification, but the difference is that instead of separating things into groups, you're trying to make a prediction on a continuous variable. So instead of separating my mice or my patients into disease and not disease, instead maybe I'm looking at the presence of a particular disease pathology. So in my laboratory, we study Alzheimer's disease. So this might be something like amyloid burden, might be something like neurofibrillary tangles, anything that you can measure and put a number on that's on a continuous spectrum. And it really anything. It doesn't have to be molecules. I know my brain goes there, but this could be body mass index. It could be, yeah, literally anything you can put a number to. And so I think I actually didn't have an example here, but something I wanted to draw your attention to is that, you know, I mentioned that classification is actually a special case of regression, almost nested within it. It's just you have 0 or 1 instead of a spectrum. And random forests are a more advanced way of doing a decision tree. Instead of making one tree, you make a bunch of them. You take averages, you do sampling, and this is definitely a case of more is better than one. You get better predictions, and you're able to do a regression instead of a simple classification. All right, great, that's fine. So really, really super fast crash course in just a few methods. And again, I'm happy to speak methods with anybody who's interested after or whenever. So you've built a model. You've applied machine learning to your data set, and you have a model, and is it any good? Is it useful? How do we assess that? So obviously I put the answer up there. But, so the first thing you want to do is you want to know if it's accurate. Does your model actually predict the thing that you want to predict? And this is very similar for you know, non-machine learning things, right? If you're trying to classify something, you want to know if you were right or wrong. And sometimes you want to know you were right or wrong in which direction. So tried and true. True positive, true negative, false positive, false negative. You want to have more things be right than wrong. So this accuracy, whether it's a machine learning method or anything else, is usually expressed as a % correct. Or if you're talking about error, as a % incorrect. Classification is really straightforward and easy to understand. Regression, though, you are almost never going to be right on the mark when you're predicting. So instead, we use something called root mean square error. This is the most common way. There's lots of ways to describe your error, but especially for linear methods, or often even for nonlinear methods, we still use root mean square error. And this is the same as for just doing a regular old univariate logistic regression in Excel. You look at where your prediction is, this line that I drew, and you look at where the actual point was, and that is your error. For all of these different points, there's some error, and you subtract out the predicted from the actual, you square it, and you divide it by the number of points you have. Take the square root. So that's pretty straightforward. Now, I'm going to ask a question, so somebody just, you know, yell it out. If I was doing a classification between two groups, what's a good accuracy? And I know it depends on the application, but just in general, what would you say, you know, if I wanted to just say I'm better than random? What would random be? Don't be shy. 50%? That's a pretty good guess. Often. Whoops, sorry about that. I don't know what happened here. Okay, sorry, I'll come back to that. If your data landscape is flat, but often in biology, our landscape is not flat. And the comparison isn't fair. So for example, even before I go to that, because that's just confusing, I'm standing here in this room. This room, I could calculate the volume of it. There's a lot of space. But there are certain areas of this space that I cannot physically occupy. I cannot hover 10 feet above where I am standing right now, according to the laws of physics, unless I have a jetpack, which I do not have. So I can't be everywhere. The space in here is not flat, even though the floor looks flat. But the volume of this room, it's not. So now let's think about a coin flip, because in biology, it's often the same way. If I have a coin, and it's an honest coin, and it's undamaged, and I flip it, random is about 50%. Now, if I took that coin, and I took some pliers to it, and I bent it in half, so that if I looked at it from the side, it looked like this, and I looked at it from the front, it looked like this, and I flipped that coin, it's going to land on the same side every single time. Now, somewhere in between there is where biology lies. So here's theoretical random. And this often is random at things like the molecular or submolecular scale. But when we're talking about cellular, tissue, certainly people biology, there are some configurations of this multi-part system that just can't exist. If my variables, for example, were height and weight, again, like my 2-year-old is about 3 feet tall and 30 pounds. However, when he grows up to be however tall, let's say 6'2", if he's 30 pounds, that's probably not physically possible. So that space is not going to be occupied, that variable space, because our variables are tied together. The more things you're looking at, and especially in biology, the less likely that your variables are independent. I study cellular signaling, and so we look at a lot of protein levels, gene transcripts, what have you. If any of you work at the molecular level, you know that like everything affects everything. Or if you study complex systems, for example, or even people, right? Everything kind of affects everything else. And so we cannot rely on the landscape being entirely flat. So how do we understand then if our accuracy is good? If we get 75% accuracy, is that good? Maybe. Is it meaningful? Who knows? So we have to test what is random. And so a very sort of basic way that we can do this is, so this is a small part of a real data set. If I have a data set that looks like this, and each row is a sample, each column is a parameter. Here it was a cytokine. And then I have my outcome. This is a regression, obviously. I can just hold this constant, keep that input exactly as it is, shuffle this, just randomly shuffle it. What I've done is I've preserved the landscape of the data, but I have made it nonsense. I've made the connection between the measurements and the outcome, I've made it nonsense. And so if I build my model and I have 75% accuracy, I've seen it, then yeah, my 75% isn't good. So what we usually do is we do this shuffle and rebuild a model like 100 times, or it depends on how many samples you have. And we make a distribution. And it's usually a normal distribution, a Gaussian distribution, bell curve, of accuracies for those models. And then we see where in that distribution did our model that we constructed, the real one, where does it fall? How good is it? How many standard deviations? And so I know we have at least 2 physicists in the room, 3, 4 physicists in the room. Normally, one would say, oh, 5 standard deviations. 100% no, that is not going to happen here in this biological data. Almost never. I actually have had it happen recently. I was very surprised. If we can get 1 standard deviation away from the mean, we'll call it useful. I don't want to use it clinically or even pretend that I could predict disease, but I'll use it for its biological insight. Absolutely. I do want to go back and talk about something that I forgot, and I'm sorry about that. So when we are talking about the accuracy, there's 3 different types of accuracy that we can mention when we're doing machine learning. The first is the calibration accuracy. And this is when we're actually building our model, or when you're doing your regression, you're actually doing the fitting of the model, and you do that root mean squared error that I showed. That is the calibration. It's when you're making the model itself. The second kind, which sometimes is the only other kind, depending on your goal, is validation accuracy. Often this is a cross-validation. It means you take part of that data set that you have and you leave it out. And you come back later and you test your model on it. So it's still from the same data set, it's been left out. And sometimes you'll do like a multi-fold, so you'll take out every single possible combination of 5 things or 10 things and remake all of the models and let them fine-tune each other. So this is a validation accuracy. This is a better judgment of the accuracy of your model, because it's an accuracy of something where you're not telling it the answer first. And it better fits your original data set because there's a lot of variation in there. If you have one really odd point and you leave it out of the model, odds are when you try to predict it, you're probably going to be wrong. So this gives you more of a real-world picture. But the actual best real-world picture is, how does your model perform in the wild? So if I were building models of something maybe that I was going to program into a device, for example, that might automatically administer a medication to a patient, that thing better be awesome, because somebody's health relies on it. So rigorously, not just the data sets that I've done in my own lab, but you would go to other data sets that don't have your hands on them. Somebody else has done it. It's old, it's with a different method, and you get that same type of data and you try to make your prediction again. And you see, does your model still predict it well? Is your model reliant on your hands, your patient population, your animal facility? So these are how you judge, okay? You have your model, how great is it? Okay. We are whizzing through this. Anybody, any questions? Thoughts? Existential crises, anything? No? Okay. So all well and good. How do you choose a machine learning method? I tore through a bunch of them real quickly. But the truth is that there is no perfect tool. There is only the right tool for your particular data set and your particular question. Just like in experimental biology or in clinical research, there's never a perfect method. So what do you take into consideration? It's mostly the stuff I've already talked about. Supervised versus unsupervised. So there's a couple questions in here, actually, and some of them involve stuff we didn't get into here. But do you have a particular outcome that you're interested in? And do you think that that outcome will have a low signal? Low signal to noise. Probably best to go with a supervised method. Do you know that you're going to have, do you want to be able to learn on super large data sets where you might have to infer things? If you're working with like these really big, large databases where you have, I don't know, 1 million N, unsupervised might be great. It can find patterns, it can learn. This would be more of the deep learning that I know that Mike talked about earlier. The more data you have, the easier it is to get a good model with an unsupervised method. Linear versus nonlinear. I didn't talk about this a lot, but some of the methods that I mentioned, like principal component analysis, partial least squares, these are linear methods. There is a matrix, there are linear relationships that are assumed both among the covariation among those individual measurements that you're making, as well as the relationship of those measurements to the outcome. Sometimes that's good enough, often. It depends on the type of data you're working with and the type of questions you're asking. I'll be honest, I rarely go past linear methods because I don't need to. Often the linear method is good enough for my objective, which I'll get to in a moment. But it's important. So if you're investigating something fundamentally nonlinear, then you probably need a nonlinear method, depending on what you're trying to predict or model. But so this is an important question and something you need to consider about your data and your question. Interpretability. I probably should have had an entire slide dedicated to this because it's important, but interpretability means, do I care why the algorithm made its decision and how? Deep learning methods are often, not always, but often black boxes. We call it a black box because we put something in, something happens, we don't know, and then an answer comes out. There's a whole field of computer science that exists to try to figure out or backtrack what's going on in some of the deep learning methods with success, but it's difficult. On the other hand, like complete other side of that spectrum, there's those linear methods that I mentioned. One of the reasons that I love linear methods is because, first of all, if they screw up, it's obvious. If you have made a mistake in your model, it's usually obvious to you because you can both understand your result, you know exactly what it's doing. It's linear algebra. You could do it with a pencil and paper, you just don't want to. Nobody wants to. But because of that, I can look at my outcome and it will also be able to tell me very plainly, look, there's a line on a piece of paper. Here's the contribution of each of those original parameters to predicting this outcome. This is important, this not so much. And often for me, that's important because if I'm trying to, let's say, so in a recent paper, we were looking at the progression of Alzheimer's disease and how inflammation progresses in these different brain regions. And to me, regressing against the age of the animal over the course of disease, I could see a signature, a pattern, like the ridges on a key. Okay, how much is this going up? How much is this decreasing with how much time? And it was predictive of disease and of disease progression and of the animal's stage in disease. And that to me reveals some sort of potentially mechanistic information that I can then test. Because at the end of the day, this is statistics, they're associations. Machine learning is, the machine learning I'm talking about is all statistical. So it's not We can, with certain methods, infer causality, but we can't prove it. So always got to go back to an experiment. So that interpretation can be really valuable. On the other hand, if you just want a complex biomarker for something, a pattern, you just want to put in a CBC, a complete blood count, somebody's lab results, and I want the machine to tell me if there's any medical problems I should be aware of. I really don't necessarily care how it got the answer. I just want it to flag that sample for me, that there might be some sort of issue that the physician should follow up on. So it could be, the answer to this could be yes or no. And interpretability is different with every single type of method. I'm going to put these two together. Caveats. How many of you do bench work in a lab? Okay. How many of you see patients? Okay. So I'll go with the lab example, first of all, because I'm more familiar with it, but second of all, because maybe I might be able to explain better why this matters. So every single time someone does an experiment, the particular type of thing you're doing has some sort of caveat. If I shine a light to try to see if there's something there, it's easier for me to see something if it's not transparent. It might be transparent, that doesn't mean it's not there. So that's the sort of relationship we're talking about, the caveats of your data. How are the measurements performed? How are the measurements performed, and how does that affect what answers you might see? And your interpretation. Then you've got to match that up with the caveats of the machine learning technique. And depending on there's any number of trade-offs. Like I said, there's no perfect tool. So that's all I have for you. I am more than happy to take any questions. This is my contact info, my stuff. Feel free to reach out in any way that is most comfortable for you. And thank you very much for listening.